everyone. I'd like to welcome everybody all around the world to this week's Global Immunotalk. My name is Laura McKay. I'm from the Doherty Institute at the University of Melbourne in Australia. And on behalf of the 2022 Organising Committee of Global Immunotalks, I would like to introduce this week's speaker. It gives me great honour and pleasure to introduce Dr. Ansuman Satpathy from Stanford University. Just before I introduce Ansu and his work, I'd like to remind you that we'll have our virtual discussions and our Q&As on Twitter after the talk, and I'll give instructions at the end of the talk on the hashtags you need to use to do so. And just to remind you to join us next week, tune in for Donna Farber, who'll be joining us from Columbia University. So Ansu, thank you so much. We're so happy that you could join us today. Just to give everybody a little background on Ansu, just found out Ansu was born in Germany. He then went on to do his undergraduate training at the University of Illinois and King's College London, and then entered an MD PhD program at Washington University under the mentorship of Ken Murphy. In 2014, completing his MD PhD, alongside his medical residency training at Stanford University, Ansu went on to do a postdoc under the guidance of Howard Chang. And it's really at Stanford where Ansu really became a leader and really well known for developing multi-omic sequencing technologies, which his group continues to pioneer. Ansu became an assistant professor in 2019. His laboratory currently interfaces genomics, computational biology, and immunology, and he's done some really impactful work most recently on the immune response to cancer and immune checkpoint blockade. Ansu is published at the absolute highest level. If you look up some of his beautiful work, you'll see it's popping up in Nature, Science, and Cell, and quite rightly so. Ansu's been decorated by numerous awards, and I had to write them down, just a few. They would take too many to say, all of them. But there's the Pew Stewart Scholar. He's a Baxter Foundation Scholar. He received the Cancer Research Institute Impact Awards, the Bill and Melinda Gates Foundation Innovation Technical Award, and the Michelson Prize for Human Immunology, amongst others. So Ansu, welcome. We're so happy to have you join us for this Immunotalk. And as you know, as part of our program, we loved for the audience to get to know you just a little bit better. And so we have a personal question for you. And what we'd like to hear from you is, what is the challenge that taught you most in your scientific career?
So thank you, Laura. I mean, first, let me say thank you very much for the opportunity to be here and share some of our work. It's really a wonderful seminar series, which we, my lab has really enjoyed over the past few years. And so it's really nice to be able to share some of our work in this forum as well. In regard to the question, maybe I could tell a short story about the beginning of my scientific career in many ways when I was a PhD student. And so, at the time, this is the late 2000s, what we were thinking a lot about was the transcriptional programming or molecular programming of dendritic cells and of that lineage. And it was very early in the field, and so people didn't understand very basic questions about what distinguished those cell types from dendritic cells from macrophages or other myeloid cells, and what was sort of underlying their programming. And so, to make a long story short, we had done a lot of work to identify a transcription factor called ZBTB46, which we predicted would be the lineage determining factor for dendritic cells. So I spent a lot of time making this knockout mouse, and had all of my hopes and dreams pinned on that being the key transcription factor. And that turned out not to be the case. So we looked at the knockout mouse once we made it after a few years and had 0 phenotype. And so, that was quite a big setback. And, I was sort of questioning, had I wasted a lot of time? Did I have a scientific future going forward? Would I even get a PhD? And it took a few weeks to sort of get over that feeling. But what came out of the other end is, in talking to a lot of colleagues and my advisor and a lot of people in the WashU community, was, could we actually leverage this tool, this mouse model, to ask different questions than we had anticipated in the beginning? And we had the foresight to knock in a GFP cassette into the locus as well. And so now using that tool, we're able to ask questions and I think come to more interesting discoveries about the early progenitors of dendritic cells, their tissue programming, what was their function in different settings, and eventually actually came to other transcription factors that were required for their development. So, I think that experience, very early on in my scientific career was important. I mean, first it taught me that more often than not, I'll be wrong in my hypotheses, and that has continued to be the case. But maybe more importantly, to be thinking about not only the sort of the first order hypothesis that you're testing, but also multiple hypotheses at the same time that maybe you could approach with the same set of experiments or the same set of tools. The second thing that it taught me was maybe more personal, was that science is hard and it takes some perseverance, and you just have to keep going at it, and you're going to hit a lot of walls on the way. And eventually, through collaboration and through talking to your community, you can find other paths that you can go down that will be rewarding as well. And so, in the process of reorienting ourselves in that project, we, struck up something like 12 or 15 different collaborations around the world to answer various different questions. And a lot of those are still ongoing. I see some of those people in the audience today. So, I think the take-home message for me was that things often don't go according to plan in science, and often the excitement and the discovery is in the paths that you wouldn't have expected from the beginning. So always be open to that, thinking about that.
I love that, Ansu. Thank you. I'm sure there's so many scientists that can absolutely resonate with what you're saying. And I think it's so important to share with the younger scientists watching today that, even the best scientists like yourself, constantly undergo failure. So I think it's really important that you shared that. Thank you.
I agree, and I would say that failure is something that happens more often than success in
absolutely
career. Yeah.
So, Ansu, we're so looking forward to your talk. If you could share your slides, we're really excited to hear what you have in store for us. Thank you.
Thank you, Laura. So, I'm excited to talk about some recent work that we've really done over the past 2 or 3 years, applying high-throughput genomics to understand cancer immunotherapy. So what my lab is generally interested in doing is developing and applying high-throughput genome-scale technologies to really understand the fundamental response of the immune system to cancer, particularly in the setting of cancer immunotherapy. We think about this at multiple levels. So we think about this at the level of what are the constituent cells in the tumor microenvironment, using things like single-cell RNA sequencing. We try and pair single-cell phenotype information with orthogonal information in the same cell. For example, T-cell receptor sequencing to understand antigen specificity or clonal lineage trace. We try and understand the regulatory mechanism, the gene regulatory mechanisms underlying particular cell states or cell phenotypes. And then we try and synthesize all of that information together to really understand the molecular wiring of a particular cell. And can we use that information to rewire the cell using things like genome engineering? And then finally, we not only think about using these high throughput tools to understand the immune response, but also to understand the cancer cells, in particular to identify cancer antigens that might be safely and effectively targeted by some of the immunotherapies that we're developing. And today I'll try and take you through actually most of these different facets of what we're doing in our work. So I think this audience needs no introduction to this topic, but suffice to say that over the past decade, we've really seen a transformation in the way that we treat cancer in patients. For many patients and many cancer types, what we now do as first-line therapy is not chemotherapy or radiation or surgery, but instead give them agents that target their immune system, so-called immunotherapies. And perhaps the cardinal example of this is checkpoint blockade, or antibodies that bind to inhibitory receptors on T cells that then reinvigorate their function to kill cancer cells, just in particular PD-1 and CTLA-4, pioneered by Jim Allison and Tetsuko Hanjo, who won the Nobel Prize a few years ago. And so what I'm showing on the bottom is a patient from Tony Ribas who had stage 4 metastatic melanoma, and their disease had really progressed despite all of the standard of care therapies at the time. This is in the early 2000s. So on the left, you can see this large lung mass in the chest cavity, and excitingly, after treatment with anti-CTLA-4 therapy, that tumor melted away, and even a decade later, that patient remained cancer-free. So examples like this have really inspired us and showed us the hope of immunotherapy, and that, when it works, it really works. The problem is that responses like this may be more of an exception than a rule in many cancer types. And so actually most patients and most cancer types do not respond in this same way or derive this amount of clinical benefit from immunotherapies that are currently being used to treat patients. So what we and many others in the field have been thinking about is, can we actually understand the fundamental mechanisms of how cancer immunity works when it is effective? And then use that information to either engineer novel immunotherapies or novel combinations of immunotherapies that could improve responses in many more patients and give this clinical benefit to more people. So we really got into this topic by studying a cancer called basal cell carcinoma, or BCC. BCC is the most common cancer in the US. So we see about 2 million new cases a year. This is a skin cancer, so fortunately you can catch it relatively early in most patients and simply cut it out, and that's an effective treatment. But when BCC becomes more advanced and goes to other organs or spreads locally, the treatment options are relatively limited. And so what we did at Stanford, really led by the effort of Ann Chang, was one of the first monotherapy trials for PD-1 blockade using pembrolizumab in advanced BCC patients. And what I'm showing you on this slide is that about half of patients actually derived a significant clinical benefit from PD-1 monotherapy alone. The useful thing about this trial, or one of the useful things about this trial, was that we could actually use these patient samples pre and post therapy to ask some very basic questions about how the immune response is working in cancer. And so again, this is a superficial cancer, skin cancer, and so the tumors are large, and you can get samples both pre and post therapy from the same site. And so this allowed us to ask some very basic questions at the time. For example, what is the diversity of T cell phenotypes in the tumor microenvironment? Is there one cell type? Are there 20? What are the phenotypes of the T cells that are going into the microenvironment? Which of these T cell types are actually responding to PD-1 blockade? Again, is it the majority of the cell types in the microenvironment that are being reactivated by this treatment, or is it a subset of them? And then importantly, what is the clonal origin of these T cells that are responding? Are they, and the prevailing view at the time was that the way that PD-1 blockade works is by binding to T cells that are within the tumor microenvironment and reinvigorating their function, rather than working in the tumor periphery. So to answer these questions, we performed paired single-cell RNA and T-cell receptor sequencing using a droplet method. And so what we did is use the patients, samples from patients in the trial that I just described to you, so 11 BCC patients, and then isolated tumor samples pre and post therapy, again, site match, so from exactly the same site about 2 months apart after treatment. We isolated the tumor biopsies, dissociated them into single cells, and then captured in this droplet format to amplify both the cDNA to get a phenotype information about each cell, and then also paired TCR alpha beta sequence. As many of you know, the TCR locus is somatically recombined during the development of T cells. And so one, TCR information provides some information or predictions about antigen specificity, but it also provides a clonal lineage information, because that recombined TCR locus is passed from parent to daughter cells. And so if you see multiple cells with the exact same TCR sequence, you can assume that they derive from the same parent cell, or they're clonally related. So what I'm showing you on the bottom left is a reduced dimensionality plot, or a UMAP, of all of the T cells from all of the patients put onto the same plot. And so what we were surprised to see at the time was really the diversity of T cell types in the tumor microenvironment. So to orient you, on the left side, we see a number of CD4 T cell types, including regulatory T cells, Th17 cells, TFH cells, for example. And on the right, we really see the full spectrum of activation and differentiation of CD8 T cells, starting from even naive cells to activated and effector cells, memory cells, and even exhausted cells. what I'm showing on the right is the same UMAP, but now colored by whether we obtained a paired TCR alpha beta sequence for each of these cells. If we did, we color the dot blue. If we didn't, it's gray. And you can see that for the vast majority of cells, we obtained both phenotypic information and also TCR alpha beta information. So using this paired information, we could now ask some of the questions that I proposed in the beginning. The first question that we asked is which of these phenotypes of T cells were actually responding to PD-1 blockade? And we used clonal expansion of T cell clones as a proxy for their activation status post-therapy. And so what I'm showing you is a clonal expansion Gini index, pre and post-therapy segregated by T cell phenotype. And so what you can see is that post-therapy in the dark blue here, really only one phenotype of cell responded to the PD-1 blockade therapy, and that was these exhausted CD8 T cells. We could start to pair this clonal expansion information with other features of the cell. For example, markers of tumor specificity like ENT-PD1 or CD39, described by Evan Newell's group, and also other markers of T cell exhaustion, including PD-1 itself. So I hope you can see, using this paired information of multiple orthogonal features of a cell, we can start to pair features of the T cell response in a tumor. So clonal expansion of the cell going into the tumor, recognizing the antigen and clonally expanding, tumor specificity, and the development of T cell exhaustion. The next question that we wanted to ask is, where are these responding T cells actually coming from? And again, the prevailing view in the field was that the way the PD-1 blockade worked was by reinvigorating cells that were already in the tumor microenvironment. The only way you can really ask that question is by having paired samples, pre and post therapy, from the same site, and some clonal lineage information, right? So you can actually ask what's happening to the cells, or were the cells there pre-therapy if they were there post-therapy. And so that's what we did. So in each patient, we compared the post-therapy and pre-therapy samples and asked, of the clonally expanded cells post-therapy that were responding to PD-1 blockade, what fraction of them were actually present in the same patient in the same tumor pre-therapy. And so what I'm showing you on the left is the fraction of novel clones, meaning cells or clones that were not present in the same tumor pre-therapy. And you can see that for most, each dot here represents an individual patient. And so for most patients, and even in some cases 100% of the time, the responding T cell clones post-therapy were not present in the same tumor pre-therapy, suggesting that PD-1 blockade may additionally work to recruit new T cells from a tumor periphery to come and join the fight against that tumor. So not only were the exhausted T cell clones pre-therapy not responding to PD-1 blockade, but we wanted to ask if they were also persisting long-term in the patient. And so to answer this question, we worked with Matt Hellman and Andrew Chow at MSKCC and looked at patients who received PD-1 blockade that had non-small cell lung cancer. And so here again, we performed single-cell RNA and TCR paired sequencing, single-cell sequencing, from about 250,000 cells. And again, you can see a lot of the same phenotypes in the lung tumors that we saw in the BCC tumors. And now we're asking, of these clonally expanded exhausted cells, how long are they persisting in that patient post-therapy compared to other phenotypes of cells? And so the way we were able to do that is by doing bulk TCR profiling in the peripheral blood and tracking these T cell clones over time in each patient. Here showing 3 patients. In one case, up to 3 years after the PD-1 therapy. And so again, a long story short is that if we ask of each T cell phenotype, are there differences in their persistence long-term in the blood? What you can see is that exhausted cells did not persist well over time. So by the 2 or 3 year mark, the vast majority of these cells, not only did they not respond from the pre-therapy sample, but they didn't persist long-term in the blood in the patient, particularly compared to other T cell phenotypes. So what this tells us is that the development of T cell exhaustion seems to be a fundamental limiting factor to their response to PD-1 blockade. Not only are they not clonally expanding in the tumor post-therapy, but they're also not persisting long-term in the blood and sort of dying out. So it seems like this development of T cell exhaustion in these human tumors is a fundamental barrier to the response of the T cell to cancer immunotherapies. And so what we wanted to understand is, what are the molecular and particularly epigenetic programs that might underlie this exhausted T cell state. And so to answer that question, we developed a droplet-based epigenetic method called single-cell ATAC-seq together with Will Greenleaf, Howard Chang, Jeff Granja, and the 10x Genomics team. And so I won't take you in depth through the method here, but what we did is adapt the ATAC-seq method developed by Will Greenleaf and Jason Buenrostro and Howard Chang to be compatible with a droplet method. And so what we're doing is we're transposing individual cells with a transposase, which tags open chromatin sites in the genome. So these are putative enhancers and promoters. And then we're loading them into this droplet generator, which then barcodes each cell and each transposed genetic loci. And so a long story short is that what you can do is, at the end, is identify open chromatin sites and single cells in high throughput in thousands of cells at a time. And so what I'm showing you here is now analysis of T cells in the tumor microenvironment from exactly the same BCC patients that I've already shown you single cell RNA-seq data for. And so what you're looking at is, again, a UMAP plot, a reduced dimensionality plot of now single cell ATAC-seq profiles. So the position of each dot represents the epigenetic state of a single cell. And so again, you can see all of the same CD4 T cell types here on the left, and then the differentiation trajectory of CD8 T cells on the right. And what you can immediately see is that exhausted T cells have a very distinct epigenetic state from other T cell phenotypes in the tumor microenvironment, just as distinct as regulatory T cells, Th17 cells, TFH cells, etc., exhibiting about 5,000 unique regulatory elements that are not seen in the other phenotypes of cells in the TME. So this tells us that exhausted cells are actually quite a distinct epigenetic state, and that probably underlies their dysfunction. It's not just the upregulation of a few inhibitory receptors on an otherwise effector T cell. And therefore, it makes sense that if you just blocked PD-1, that entire epigenetic landscape is already there mediating the dysfunction. Just by blocking one inhibitory receptor, you're not likely to reverse all of that dysfunction. And this supports work from Andrea Schiedinger and John Wary and Nick Haining and Arlene Sharp and many others showing a distinct epigenetic states of exhausted T cells in other settings as well. Importantly, we now looked at this data set and segregated cells by cells that were derived pre and post therapy. And so what I'm showing you is the same UMAP, but now the cells, or the single cell ATAC-seq profiles, are colored by whether the cell was derived from a pre-therapy or post-therapy sample. And if they're post-therapy, they're in blue. If they're pre-therapy, they're red. And so again, what you can immediately see is that the exhausted T cell epigenetic profile is not changed by PD-1 blockade. The cells enter exactly the same epigenetic state. So what I've shown you in the first part of the talk is that a large number of clonally expanded CD8 T cells post-therapy are not found or not expanded in the same tumor pre-therapy, suggesting that a major mechanism of how PD-1 blockade works is not by reinvigorating the exhausted T cells that are already in the tumor, but instead by expanding a novel cohort or a repertoire of T cell clones, perhaps from the periphery of the tumor. I've also shown you that exhausted T cells have a distinct epigenetic state, which perhaps mediates their dysfunction. And we view this epigenetic state and the fact that it's not reversed by checkpoint blockade as a significant barrier to long-lasting immunotherapy. So what we've been thinking about over the past few years is can we reverse or prevent or extend the window of time in which a T cell develops this exhausted state? And so to answer this question, the first thing that we did is ask a very basic question, which is, do all T cell clones that respond to chronic antigen actually become exhausted or develop this terminally exhausted epigenetic state? So what we did is we worked with Takeshi Egawa's group at WashU in St. Louis to analyze a large or generate a large data set of paired single cell RNA and TCR sequencing information in the setting of chronic viral infection, in the setting of LCMV clone 13 infection compared to an acute infection with the same virus. And so I'm showing you in the top left here is about 200,000 single cells. Again, the data is paired RNA and TCR-seq information. And the colors represent the different phenotypes of cells, either obtained from mice that were infected with an acute infection with LCMV or a chronic infection with clone 13. And what you can see on these right plots is the TCR-seq clonal expansion information. And so what you're looking at is in acute infection at day 8, so early in infection when the virus is still around, you see clonal expansion of these cells that have an effector T cell phenotype. And then by day 21, when the virus is mostly cleared, you see the development of memory-like T cells. So this is what we would expect. In contrast, in the setting of chronic infection, so this infection is not cleared by day 21, what you see is the development of very distinct T cell phenotypes. And now through the work of Rafi Ahmed and John Wary and many others, we know many of these subsets and their roles in the development of T cell exhaustion. And so what you can see is a progenitor exhausted-like cell expressing TCF1. You can see this intermediate cell, which has recently been described to express CX3CR1, and then the development of terminal exhaustion with high levels of PD-1 and other inhibitory receptors. What you can also see is clonal expansion of a distinct subset of T cells, which I'll come back to. So now looking at clonal information, what I'm showing you on the bottom is individual T cell clones. So every bar here represents an individual T cell clone. So all of the cells have exactly the same TCR alpha beta sequence. And the color of the bar represents the phenotype of that cell. And so you can see in acute infection, we can see large amounts of expansion of these top 10 T cell clones. And at day 8, most of them are this effector T cell phenotype. By day 21, all of these clones develop into effector memory or memory T cells. What you can see in contrast in chronic infection at day 8 is that early on, you have the development of progenitor exhausted or these transitory exhausted cells expressing CX3CR1. And then by day 21, a lot of these clones develop into terminally exhausted cells, so these red bars. What we were surprised to see is a number of clones which did not follow this same pattern. So instead of going down this purple, green, and red terminal exhaustion path, what they did was develop into these effector-like cells that is this population of cells that I pointed to before. So if we zoom out now and ask about the global patterns of differentiation trajectories, so looking across thousands of T cell clones, again, this is a polyclonal response to chronic infection or chronic antigen, what we see is 3 predominant patterns of T cell differentiation. So we see this pattern that has been described before in the field where cells develop into progenitor exhausted cells, which perhaps self-renew and maintain the proliferation of these cells. Developing into transitory exhausted cells, and then finally ending in terminally exhausted cells. However, this is only about 50% of all of the clones that we've tracked. The other 50% go down different differentiation trajectories, largely including the difference is largely that they include these effector memory-like cells. So either they develop into both terminally exhausted and effector memory cells, so you can see an example of a clone like that up here, or they develop only into these effector memory-like cells. And we're still trying to understand the difference or the reason that a polyclonal response would develop these different pathways, but we have evidence to show that these effector memory-like cells, which we're calling KLR cells because they express large amounts of killer-like receptors and other effector molecules, have an increased propensity to perform cytotoxic functions and also to develop memory. Whereas these clones that go down a terminal exhaustion path primarily have the ability to persist or enter high antigen niches. So we think that balancing these 2 phenotypes or these 2 functions of the cells might be beneficial to an organism to develop in the setting of chronic antigen. We're still trying to understand, again, the regulation of how these different paths are developed for each clone. We have some evidence that it is driven by TCR affinity. So in a number of experiments, we're either testing the TCR affinity individually of particular TCR clones, or in a simple experiment where we take cells that are staining very strongly for a tetramer, sort of, again, a proxy for high TCR signal strength versus lowly. We can see that low affinity TCRs preferentially develop into these effector memory-like cells in the setting of chronic exhaustion, whereas high affinity cells will are more likely to develop into this terminally exhausted pathway. So all of this tells us that in the setting of a polyclonal response to chronic antigen, not every T cell clone that sees that chronic antigen will go down this terminal exhaustion path. And so this gave us some inspiration for the idea that perhaps we could achieve the same thing genetically. Perhaps we could find, again, non-TCR signal strength factors that we could perturb in a cell to make that cell persist longer in the setting of chronic antigen, or simply not become or as quickly become a terminally exhausted cell. This could be relevant not only for the setting of checkpoint blockade, but also for cell therapies. And as many of you know, the second pillar of cancer immunotherapy, in addition to checkpoint blockade or antibodies targeting inhibitory receptors, is the development of cell therapies, and in particular, the development of chimeric antigen receptor T cells or TCR expressing T cells. And so very simply, what a CAR T cell is pairing the antigen recognition domain of a monoclonal antibody against a desired antigen specificity with the intracellular signaling domain of a T cell receptor, thereby making a chimeric antigen receptor that can recognize an antigen of choice and signal the T cell to activate and kill that antigen-bearing cell. So you can isolate T cells from the circulation of a person, endow them with this chimeric antigen receptor, and then infuse them back into the patient to reorient those T cells to recognize the cancer cells in that patient. We worked with Crystal Makel's group to show that CAR T cells in many settings, both in vitro and in vivo, develop features that look very similar to T cell exhaustion. So in some settings, they don't expand as well, either in culture or in the humans after infusion. They upregulate a number of inhibitory receptors, and even epigenetically, by looking at ATAC-seq or chromatin accessibility data, we see a lot of similarities between CAR T cells that are becoming exhausted and the exhaustion that we see in the setting of checkpoint blockade, for example, in BCC, as I showed earlier. And this is also supporting really beautiful work from Anjana Rao and many others doing the same comparisons. Another collaboration, this time with Carl June's group and Ed Stadmauer, we wanted to ask, could we actually do something about this? So was there a path where, let's say theoretically, we could identify genetic regulators of exhaustion? Could we actually engineer cell therapies to knock out those factors or overexpress those factors and put those back into patients? And so what we were very fortunate to work with Carl's group on was the first in human Crispr trial in the context of engineered T cells, in this case, NY-ESO TCR-bearing T cells. And so the whole goal of this trial was to show that we could take T cells from a patient, engineer them using CRISPR-Cas9 ribonucleoproteins, in this case targeting PD-1 or the endogenous TCR, edit them ex vivo, and then infuse them back into the patient and show that this was a viable therapeutic path and that it would be safe for patients. And so again, to make a very long story short and to summarize the work of a lot of people really led by Ed and Carl, this path of therapy was safe in the patients. And so now taking all of this together, we've shown that T cell exhaustion seems to be a limiting factor for the persistence of T cells and the function of T cells in the setting of cancer immunotherapy. We've shown that this is relevant to both checkpoint blockade and cell therapy modalities. And we've shown that we can actually modify these cells using CRISPR engineering to make them function in an improved manner for patients. And so I think all of this sets up the idea that if we could identify genetic regulators of exhaustion, that might be effective as a therapeutic strategy, and that it might be able to be translated very rapidly using these things that we've done collaboratively with Crystal and Carl. And so that's what, inspired by all of that, what we decided to do was perform genome-wide CRISPR screens in the setting of T cell exhaustion to identify factors that if we deleted them would improve the persistence or function of T cells in the setting of chronic antigen. And so we've done this in multiple ways, but I'll show you two of them here. The first is an in vitro model where we isolate CD8 T cells from a mouse and then essentially try and mimic the chronic antigen that it would see in the setting of a tumor by just doing chronic stimulation in a dish with anti-CD3 and IL-2. The second model is now in vivo, and so taking CD8 T cells, editing them with CRISPR-Cas9, and then injecting them into a tumor and asking which edits actually not only resist exhaustion, but allow the T cell to do all the things that it needs to do to persist in a setting of a tumor. So in some cases, this is a higher bar than the in vitro system. In vitro, all the genetic edits need to do is improve the persistence of the T cell in the setting of chronic antigen. In vivo, the T cell needs to do much more. The T cell needs to find the tumor, infiltrate the tumor, overcome the tumor suppressive factors, and then eliminate the tumor and persist in that setting over time. So using these sort of parallel screens, we're able to first perform genome-wide screens and then more and more iteratively targeted screens to identify factors that would improve T cell persistence in the setting of chronic antigen. So the first piece of data that I'll show is demonstrating that the in vitro system that we developed, and really this was pioneered by Santosh Vardhana at MSKCC, really did recapitulate a lot of the features of exhaustion that we saw in BCC or LCMV or other settings. And so what I'm showing you here is a time course of attack-seq data in the setting of in vitro chronic exhaustion, chronic stimulation, compared to in vivo data from mice. And so what you're looking at is the accessibility. Every row in these heat maps is a different genetic element, and the heat is how open that element is. And so what we're looking at is a subset of about 3,500 regulatory elements that are somewhat specific for terminally exhausted cells. So this is the epigenetic state of terminal exhaustion. And hopefully what you can see is that over the development or over the time course of chronic stimulation in vitro, we are able to recapitulate a large amount of the epigenetic footprint of exhaustion in this setting. So using that system now, we performed genome-wide screens. And what I'm showing you on the left is around the top 100 hits that came out of the screen. And so what I'm showing you is a protein-protein interaction network of all of the hits. So just to organize the hits in a way that are understandable. And each circle represents a hit or a gene. And if that gene is colored red, then that means that deleting the gene was good for the T cell. It made it last longer in the setting of this in vitro chronic stimulation. If it's colored blue, it means that deleting it made it worse. And so what you can see is that you get this large enrichment of TCR signaling factors. This is in some ways a positive control in this screen. So again, the exhaustion is being induced by an anti-CD3 antibody. And so therefore, if you knock out any of these TCR signaling factors, that should resist that stimulation and therefore allow the cell to last longer. So this is a positive control. In addition to TCR signaling genes or proteins, what you see is a number of other categories of factors. You see cell adhesion receptors, you see mRNA processing factors, you see a lot of things involved in metabolic state, mitochondria, lipid biosynthesis, and then you see this large enrichment of factors that are somehow categorized as epigenetic factors, either transcription factors, histone modifiers, or also enrichment in nucleosome remodeling complexes, in particular the INO80 complex and the SWI/SNF or BAF complex. Interestingly, what we didn't see is enrichment of factors that delete inhibitory receptors, for example, PD-1. And again, this supports the idea that by deleting PD-1, you don't reverse the epigenetic state of exhaustion, and therefore you're likely not to increase the persistence of the cell in the setting of chronic antigen. Taking these top hits, we now perform sort of iterative in vivo screens. And so now what I'm showing you on the right is the same set of factors, but now colored by whether they were beneficial to delete those factors in vivo. So again, this is a much higher bar, right? The T cell needs to do many more things in vivo to actually successfully find the tumor and then persist in that setting. And so now what you can see is that many of these TCR signaling factors are no longer hits, right? Again, this is now a negative control. You need your TCR signaling to be able to identify the tumor antigen, in this case, ovalbumin, to recognize the antigen, infiltrate the tumor, and persist. So again, this was good to see. But now what you can see is really the enrichment of hits in these epigenetic categories. So again, hits in the nucleosome remodeling complexes, INO80, some transcription factors, and a large number of hits in the SWI/SNF complex, in particular ARID1A, SMARCC1, and SMARCD2. So next we wanted to ask, what is deletion of these individual genes actually doing to the T cell? How is it making the T cell last longer? First of all, and then second of all, are these T cells actually functional? Are we just making them persist longer, but now all of a sudden they're not able to fight the tumor as effectively with effector molecules, etc. What I should mention is that the way that we designed the screens was to look for factors that were not only specifically improving the function of the T cells in the setting of chronic antigens. So we did counter screens in the setting of acute stimulation to make sure that the knockouts were not making the cells secrete less cytokines or proliferate less in the setting of acute stimulation. So we had some confidence that these factors were doing something fundamentally important to the cell to allow it to last longer, but also maintaining their effector function. So to ask this question, we performed an in vivo perturb-seq experiment. So what we're doing here is we're taking a mini pool or micro pool of some of our top hits, including and focusing on these chromatin remodeling factors, knocking out these genes in T cells, infusing them into tumor-bearing mice, and then sorting the T cells back out at day 15 when we would expect there to be T cell exhaustion. And then performing single-cell RNA-seq paired with the amplification of the sgRNA that each cell receives. So in this way, we could pair the phenotype of the cell with the perturbation that we gave the cell. So what I'm showing you in this bottom heat map is now gene expression data segregated by each sgRNA identity. So you can see the comparisons of several of the hits, in particular these SNF factor BAF complex members here. And so what you can see is immediately a couple of things. One is that knocking out the BAF complex members had a large transcriptional effect on the cell. About a few thousand, 2,000 or so genes were changed. And if we looked carefully at those genes, we could see a number of patterns, that there was less expression of terminal exhaustion-related genes, in particular inhibitory receptors. There was an increased expression of progenitor exhausted or other stem markers, and there was actually an increased expression of effector molecules, in particular effector cytokines and the transcription factors IRF4 and BATF. We next did some in vivo transplant experiments to show that these knockout T cells actually still perform better in terms of killing the tumor. They hadn't lost that function. And so what I'm showing you on the bottom is now the outgrowth of colon adenocarcinoma line in the setting of no transplanted T cells, transplanting T cells that contained a control guide, or transplanting T cells that contained a guide sgRNA targeting ARID1A as just one example. And so what you can see is the tumors grow out in the setting of no T cells recognizing the tumors, transplanting some wild-type tumors or controlled T cells controls the tumor a little bit, but then by injecting ARID1A knockout T cells, we can quite significantly improve tumor immunity for those tumors. And finally, we wanted to really understand the programming of the BAF complex. So we had a number of hits that showed up in the SNF complex. We really wanted to understand the extent to which BAF complexes and the mechanism by which BAF complex and knocking out BAF complexes might improve the function of exhausted cells or prevent exhaustion in these tumors. And so to ask that question, we performed a series of mini pool CRISPR experiments where we're targeting each of the 29 members of the SNF complex, so it's in different SNF subunits. And so as many of you might know, there are different conformations or organizations of these subunits that make different functional BAF complexes in the cell. CBAF or canonical BAF, PBAF, and non-canonical BAF. And what we're showing you here on the bottom is a cartoon image of all of these subunits organized according to their modules, and then coloring them by whether or not they were hits in these iterative in vivo screens. And so if they're colored red, deleting that gene made the T cell function better. If they're colored blue, it didn't. And so you can see now some interesting patterns where primarily knocking out canonical BAF complexes, in particular ARID1A, ARID1B, but also some in the PBAF complex, ARID2, improved T cell function, but that was not a general rule for all BAF complex subunits, in particular for this ATPase module. So what we think is happening is that, and I think there's a lot of work to be done here, but what we think is happening is that what we're doing by deleting some of these subunits is tuning the amount of canonical BAF complex on chromatin. And so if you're missing or have a decreased amount of this canonical BAF complex on chromatin, you can't acquire this epigenetic state that I showed you earlier that is associated and perhaps underlying the dysfunction of exhausted T cells. We can show that using ATAC-seq data here on the right, so showing you a number of regulatory elements that are either more accessible or less accessible in ARID1A knockout T cells. And again, the pattern is that you have less accessibility of terminal exhaustion associated regulatory elements, and have increased accessibility of sites that are preferentially bound by BATF and IRF4 factors. So what I've shown you in the second part is that using, again, paired TCR RNA-seq analysis and single cells, we're able to identify divergent exhaustion pathways in the setting of chronic antigen. Not every T cell clone in a polyclonal response that sees chronic antigen will go down this terminal exhaustion path. And then we've been inspired by that to perform genome-wide and multi-omic Crispr screens and have identified chromatin remodeling factors as novel targets for improved T cell persistence in the setting of chronic antigen. And then finally, we've shown that epigenetic perturbation, in particular by knocking out BAF complex members, increases perhaps the frequency of progenitor or effector-like exhausted cells and improves, importantly, the anti-tumor T cell response. So just in the last 5 minutes or so, I want to switch gears and talk about how we've been using a lot of these same methods, not only to understand T cell programming, but to understand, in particular, cancer antigens that are expressed by cancer cells that might be safe and effective to target with some of these engineered cell therapies. And we really came to this, again, through a collaboration with Carl June's group. And what we were thinking about at the time, or what we had observed, was that many of these many patients who were receiving CAR T cells that were specific or targeting CD19 as a B cell antigen, these are patients with leukemia or lymphoma, there seemed to be a high rate of neurotoxicity. And this ranged all the way from mild neurotoxicity, things like delirium and aphasia, and some patients really was quite severe, progressing to cerebral edema, hemorrhage, and even in some patients' death. What we were struck by was the idea that neurotoxicity was observed in patients receiving CD19-directed immunotherapies, CAR T cells, but also bispecific T cell engagers or other sort of antibody-directed therapies. But it wasn't seen as frequently in patients who are receiving CD20-directed agents or CD22-directed agents, even though those should have been on the same cancer cell population. And so we wondered, could there be something about the expression of CD19 that was underlying the increased frequency of neurotoxicity and perhaps contributing to the neurotoxicity that we know is caused by cytokine release syndrome? So could there be sort of two things happening in CD19-directed therapies that could be contributing to the increased rate? At the same time, what we and many others in the field were doing on the genomic side was developing a human cell atlas of cell types and gene expression across the whole human body, right? So we're using things like single-cell RNA-seq to identify all of the cell types, constituent cell types in the human body, and also all of the genes that were expressed in each of those cell types. So it seemed to us that we could leverage, and that the community could leverage this a very useful resource that many people have been contributing to really understand expression of target antigens, cancer target antigens that we're going after using some of these immunotherapies. So that's what we did, and we asked very simply, was there a cell in the brain that was not a B cell or was not a cancer cell that expressed levels of CD19 that perhaps was also inadvertently being targeted by these CD19-directed therapies? And so what I can tell you is that, so what we're looking at here is a public data set from Zhang et al. Of brain cells from the human prefrontal cortex, so single-cell RNA-seq data. And what we're doing is subsetting the non-neuronal cells. So neuronal cells did not express CD19. But if we looked carefully at these non-neuronal cells, we could find a small population of cells that was not, that didn't have other B cell markers. So it wasn't classified as a B cell. It wasn't classified as a cancer cell that expressed high levels of CD19. It's about 0.5% of all the cells in this data set. And then if we looked a bit further, we could identify other markers on these cells that identified them as the cell type called pericyte. And I should mention this was a really beautiful collaboration, close collaboration with Carl June's group, Avery Posey, Dennis Migliorini at the University of Pennsylvania. So what are pericytes? And we started to learn a little bit about pericytes, and what these cells are cells that wrap the endothelial cells surrounding capillary vessels. So they maintain the tight junctions that form these blood-brain barrier and maintain this blood-brain barrier integrity. And so what studies in mice from Ben Beres's group and many others had shown is that if you genetically delete or deplete pericytes, what you get is vascular leakage, blood-brain barrier, loss of blood-brain barrier integrity, loss of tight junction stability, and you get a lot of leakage of proteins in the circulation into the brain parenchyma. So this started to make sense. Perhaps CD19 was expressed for some reason on these pericytes surrounding brain blood vessels, and by targeting them with cell therapies, you might lead to some vascular permeability that could synergize with cytokine release syndrome and other things happening in a patient with cancer, leading to increased rate of neurotoxicity. For this to really make sense, this would have to be a CD19 specific phenomenon and not seen in CD20 or CD22. And so to answer this question, we worked with Arnold Krigstein and Aparna Bhaduri at UCSF to really leverage quite an amazing single cell data set they had generated of the developing human brain. So taking different stages of development, performing single cell RNA-seq, and identifying all of the cell types as they were emerging during development. So what I'm showing you on the top left here is all of the cells that are somehow related to progenitors or end-stage cells related to vasculature, including pericytes. So we focus on these dark blue clusters here, which include pericytes, but also other cell types that wrap endothelial cells in larger and larger vessels that are sort of broadly classified as mural cells. So this includes pericytes. It also includes vascular smooth muscle cells, which are sort of the larger, the cousin of pericytes that wrap larger vessels in the brain. And so what you can see on the bottom is now expression of these genes that we've been talking about, and you can see expression of CD19 in really multiple mural cell lineages. And essentially, as soon as the cell type develops in the brain, they are expressing CD19. And there's some interesting developmental biology behind that related to the shared expression of B-cell transcription factors in the development of this lineage, particularly in the brain, so not expressed in pericytes and other tissues. But interestingly, what we don't see is expression of CD20 and CD22. So this does appear to be a CD19 specific expression pattern. And then finally, we worked with Avery's group and Dennis Migliorini to perform mouse experiments to show that infusion of CAR T cells that recognize CD19 in the brain could lead to, was sufficient to lead to vascular permeability in mice. And so what I'm showing you on the top is now mouse single cell RNA expression. It's worth noting that pericyte expression of CD19 in mice is much less than what we see in humans, and perhaps this is a reason why neurotoxicity was not flagged in earlier preclinical studies. But nevertheless, you can see CD19 expression in murine pericytes. And then importantly, if you inject CAR T cells recognizing murine CD19 into an NSG mouse where there are no B cells, so the only thing it should be able to recognize is pericyte CD19, you can induce vascular leakage of this Evans blue dye into the brain parenchyma if you're targeting murine CD19, but not if you're targeting human CD19 as a control. And this seems to be worse if you incorporate a stronger signaling domain, such as CD28 versus a 4-1BB domain. And I think it's important to note that this is, since that time, we've started to look at more and more cancer antigens that are being targeted in the field. And this does not seem to be just a CD19 problem. So we can now see expression of cancer antigens on unintended tissues in multiple, for multiple cancer antigens that are currently being targeted in humans. And so, for example, ROR1 is expressed in human lung alveolar cells. CD33 has off-target expression. BCMA has expression in the striatum of the brain. And we were interested to see this study from a group at Mount Sinai showing that patients who received a BCMA targeting CAR also had targeting in the brain of these striatal neurons, leading to a Parkinsonism-like syndrome. And then, for example, CD123, which has been being targeted in leukemia, also has expression in endothelial cells, and those patients also see some evidence of vascular leakage. So what I've shown you in this last part is that single cell analysis can identify expression of CD19 in a rare population of brain pericytes, and we think that targeting pericyte parasites that are expressing CD19 could contribute to neurotoxicity in patients who receive these immunotherapies. I think there are many open questions. There's a question of how much of neurotoxicity is driven by cytokine release syndrome, which we know contributes to neurotoxicity, versus parasite expression. Is there a synergy there in particular patients who might have high parasite expression of CD19 that synergizes with CRS? Is there heterogeneity in patients? Does this change with age? I think there are many open questions still to be answered. But importantly, I think what all of this data tells us is that we should be using single-cell genomic approaches in a lot of the databases that are being built by the genomics community to understand cell type specific and cancer specific antigens that we should be targeting or should not be targeting using immunotherapies. And perhaps that might lead to an unbiased nomination of novel antigens that we could now target for a more safe and effective therapy for patients. So thank you very much for having me in the seminar series. It's really a pleasure to share our work with the community. And hopefully what I've been able to show you is that what we're really trying to do is go all the way from understanding sort of basic principles of cancer immunity and cancer immunotherapy in patients using clinical samples, understanding sort of fundamental limits of T cells or other cells that are limiting the efficacy of these therapies using immunological tools, and then using genomics and principles from computer science to hopefully engineer improved forms of immunotherapies that we can put back into patients and then iterate on this cycle to really develop next generation therapies that could be beneficial to more patients and more cancer types. So I'll stop there and just end by thanking really my wonderful lab, many of whom I've tried to call out throughout the talk, but have really contributed to many of the stories that I presented today. And we've been really fortunate to have wonderful collaborations sort of across the global immunology community, and really fortunate to have wonderful funding agencies supporting our work as well. Thank you very much.
Thank you, Ansu, for sharing all those wonderful stories with us. I'll just now share my screen to let everybody know how they can ask you questions. So Ansu's kindly agreed to do a virtual Q&A with us on Twitter. You can either do this right now if you're joining us live, or in your own time zone if you're watching this talk on YouTube whenever suits you best. Ansu will continue to answer questions for a little while. So if you search for the account Global Immunotalks on Twitter, find the tweet that says "Ask questions for Dr. Ansu Satpathy here," and then reply to that tweet with your question mentioning #GlobalImmuno. And Ansu will be tweeting his replies from his own personal account at Satpathology. So that just leaves me to also remind everybody to tune in next week. We'll have Donna Farber joining us from Columbia University. And so thanks to everybody for joining us again today. Ansu, thank you so much for being a part of our seminar series. It was such a pleasure to have you, and we were so excited to hear your work. I'm sure there's lots of questions for you. Thanks again, and we'll see everybody next time.